Hello all, welcome to this video on antihistaminic agents. Antihistaminic agents are those agents popularly known as anti-allergic drugs. They are those drugs that are taken when we have a running nose, sneezing, nausea, vomiting, motion sickness or any allergies for that matter. So some of the popular brands that you all are familiar are Avil, that contains phenyramine malleates, cetirizine, Phenergan, Benadryl, etc. So antihistaminic agents are agents that inhibits the interaction or action of histamine at histamine receptors, especially H1 receptors. So before seeing what are antihistaminic agents, we will see what are histamines, its chemistry, biosynthesis, what are the different types of histamine receptors present in our body, what are the physiological roles of histamine, etc. The word histamine is derived from two words. One is Histos and the other is amine, meaning it is an amine present in tissues. It is a natural constituent of the body. It is a local hormone, otherwise we call it as an autocoid. They are the part of our immune system and they are mainly present in mast cells and basophils. They are seen in large numbers where the external environment comes in contact with the internal milieu. That is the cells of epidermis, that is in skin, in the gastric mucosal cells, then in the bronchi of the lungs, urinary tract, etc. They are also present in central nervous system where they act as a neurotransmitter, and in gastrointestinal tract it acts as an acid secretagogue, that is it increases the acid secretion. Coming to the chemistry of histamine. So what you see on the screen is the structure of histamine. You can see that it contains 3 nitrogen atoms. So nitrogen containing compounds are basic in nature. So histamine is a basic compound. So out of the 3 nitrogen atom, first nitrogen atom forms a part of the amino group. It is a primary amino group. And the other 2 nitrogen atoms are present in the heterocyclic aromatic ring system. And the heterocyclic ring present here is imidazole ring. And the primary amino group is connected to this imidazole ring by a ethyl side chain. And here the point of attachment of this ethyl side chain is at the 4th position of the imidazole ring. So if you are naming the histamine, it is 4,2-aminoethyl imidazole, or else you can name it as beta imidazole ethylamine. It is beta imidazole because the carbon atom carrying the amino group is alpha, and so the other carbon atom is beta, so it is beta imidazole ethylamine. Coming to the biosynthesis of histamine. Histamine is biosynthesized in the Golgi apparatus of mast cells and basophils. And the precursor of histamine is L-HISTIDINE. It's a naturally occurring amino acid and it is formed by the decarboxylation of L-HISTIDINE. And the reaction is catalyzed by the enzyme L-HISTIDINE decarboxylase. Decarboxylation means removal of carboxylic acid. And the reaction is catalyzed or carried out in presence of pyridoxal phosphate, vitamin B6 phosphate, to form histamine. Point regarding the structural features of histamine, the 2 nitrogen atoms present in the imidazole ring. One is named as pros. Prox means proximity to the amino group, that is, it is near to the amino group. And the second nitrogen atom of the imidazole is named as tele because it is far away from the primary amino group. And these 2 nitrogen atoms of the imidazole ring and the primary amino group, that is 3 nitrogen atoms, are important for interaction with the receptors of histamine. The histamine that is formed inside the mast cells and basophils are stored in its inactive form in complexation with certain proteins. Example, heparin in mast cells, chondroitin in basophils. So these histamines are released by exocytosis from the granulocytes in response to various stimuli. The stimuli can be either an immunological response that requires an antigen-antibody reaction. Degranulation of histamine storage cells takes place, and along with histamine, certain other inflammatory mediators like prostaglandins, leukotrienes, platelet activating factors are also released. Then histamine can also be released by certain non-immunological reasons. Example like certain chemicals like drugs, peptides, venoms, certain organic bases like amides, amidins, and alkaloids, antibiotics, tuberculin, succinylcholine, radiocontrast media, plasma expanders, etc. can also release histamine from these storage granules. Next, we'll see how histamine produces their physiological actions. They produce their physiological action by interacting with histamine receptors. 4 different histamine receptor subtypes have been identified in humans. The first 2 are more important, and they are named as H1 receptors and H2 receptors. And other 2 receptors are H3 receptors and H4 receptors. We'll just see the distribution of these receptors and what are the different physiological changes they bring about when histamine interacts with these receptors. First, we'll start with H1 receptors. H1 receptors are found throughout the central nervous system, and they are particularly densely concentrated in hypothalamus. In central nervous system, histamine acts as a neurotransmitter. They increase the wakefulness. So that is why when antihistamines are taken, sedation occurs as a side effect. Next is the H1 receptors present in the smooth muscles of respiratory tract, gastrointestinal tract, uterine tract, uterus, etc. And interaction of histamine with H1 receptors present in these smooth muscles causes contraction of these muscles. For example, the contraction of smooth muscles of bronchi causes bronchoconstriction. Next is the H1 receptors present in the blood vessels. The interaction of histamine with H1 receptors in the endothelial cells of the blood vessel causes release of nitric oxide and prostaglandin I2, resulting in vasodilatation, increased capillary permeability. Vasodilatation of smaller vessels like arterioles, capillaries, and venules takes place, and as a result of that, increased capillary permeability occurs, and that may lead to edema, pruritus, pain, hypotension, that is fall in BP occurs. Whereas in interaction of histamine with H1 receptors present in smooth muscles of larger blood vessels causes vasoconstriction. Then H1 receptors are also present in WBC, neutrophils, eosinophils, monocytes, dendritic cells, T cells, B cells, etc. And as a result, they have a modulatory role on the cellular immunity. Next is the interaction of histamine with the H2 receptors. These H2 receptors are present mainly in parietal cells in the gastrointestinal tract, vascular smooth muscles of respiratory tract, heart, uterus, etc. The most important and the significant effect of histamine on H2 receptors is that they mediate gastric acid secretion. That is, histamine acts as an acid secretagogue, increases the acid secretion. And the next important action of histamine on the H2 receptors present in heart is that they produces positive ionotropic and positive chronotropic activity. That is, they increase the force of contraction as well as increase the heart rate. And the increased vascular permeability is actually a combination of action of histamine on both the H1 receptors and the H2 receptors. So they produces increased vascular permeability, and as a result of that, hypotension, fall in BP can also occur. These are the action of histamine on H1 and H2 receptors. So as far as antihistamines are concerned, they refers to the blockade of action of histamine on H1 receptors. We will see individual antihistamine compounds in the next video. Thank you.